Hello, my name is Hannah Watkins, and this study is investigating the preparation, characterization, and antimicrobial activity of 6-ethoxy 6-phenyl 6-H-chromino 3,4-B quinoxaline, which we will call chrominoquinoxaline. There are two major parts in the chrominoquinoxaline compound, chromain and quinoxaline, which we can see here in figures 1 and 2. Studies show that chrominones have anti-cancer properties, and quinoxalines have antifungal and antibacterial properties. In this study, we are finding ways to combine chrominones and quinoxalines to form chrominoquinoxalines. And we want to determine if there is any combined activity or new applications. In figure 3, we have a line reaction. We started with 3-hydroxyflavone and reacted it with an alcohol and a catalytic amount of copper bromide at room temperature for 1 week. This gave us 3-hydroxy, 2,3-dioxy, 2-phenylchromane for a none, or our chrominone. Here, we have a proton NMR of the chrominone. I have the peaks labeled with corresponding hydrogens on the structure. We see a lot of peaks downfield, or to the left. And this is because most of the hydrogens are on a ring, but there are some hydrogens that are not on a ring. For example, the hydrogen labeled A. So these peaks are further upfield or to the right. Here, we have a single crystal X-ray structure of the chrominone. The oxygens are in red, there's a ring on each side, and there's a hydrogen in white, which is a part of the alcohol group. Going back to the line reaction, the chrominone was reacted with phenylene diamine in the presence of copper bromide and chloroform at 84 degrees Celsius to yield our chrominoquinoxaline. And here we have a proton NMR which confirmed the synthesis of chrominoquinoxaline, and I have the peaks labeled with the corresponding protons. For the biological experimentation, an antimicrobial susceptibility test was conducted on E. coli on TSA plates. 4 concentrations of the chrominone, dissolved in DMSO, were pipetted onto sterile paper disc and placed on the inoculated plates. In figure 7, we can see each section labeled. 1 through 4 are the concentrations from most concentrated to the least concentrated. And 5 is the negative control, which was the DMSO solvent only. In figure 7, we do not see any antibacterial activity. But in figure 8, there was some antibacterial activity. In figure 8, 3 replications of the 50 milligrams per milliliter concentration was tested. This was the highest concentration. It was tested along with the DMSO negative control. The asterisks indicate the disc with the chrominone. Around each of these discs, we see a clear zone. This indicates antibacterial activity. The main difference between figures 7 and 8 is that figure 7 has 15 microliters of the concentration, and figure 8 has 30 microliters of the concentration. This indicates that volume played a role in the antibacterial activity here. The chrominoquinoxaline derivative that was synthesized was not soluble in any suitable solvents for biological testing. The derivative that was made was not very polar. This is most likely why it did not dissolve in any of the solvents that were used. But having biological activity in the chrominone is an indication that there will be activity in the chrominoquinoxaline. This study is ongoing, and in the future, we plan to use reagents to synthesize structures that are more polar, such as the ones in figure 9. This is in hopes to improve its solubility. We also plan to test the derivatives against other bacteria, fungi, and cancer cells. We'd like to thank Georgia Southern University Biology and Chemistry and Biochemistry Department. And we'd like to thank you for watching.